Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12th Molecular Basis of Inheritance Part 9, discuss the transcription unit and in the genes. A gene is defined as in a functional unit of any inheritance. So gene, it is in a part of the unit of inheritance characters. So there is no the ambiguity, that is gene is located on the DNA, because the genes are present in where? Genes are present on the in a locality of the DNA. It is difficult to the literature that defined as in a gene terms, that is in a terms of the DNA sequence. The total DNA sequence, the DNA contains as in what? The number of the codons as present, the total codons they are having what? They providing as a proper information. Correct? Let us. The DNA sequence coding for the tRNA or RNA molecule is defined as a gene. So, for example, here I took here one DNA strand. Here I take completely one DNA strand. So, where the DNA will be there contained as an mRNA, where the DNA contains mRNA as well as any rRNA. So, the genes are called as what? That is called as a gene. That is called as what? Gene. However, to define as in the cistrons, the segment of the DNA coding the polypeptide, the structural genes in the transcription unit that could be there said to be as a monocistronic. That mostly you see as in a completely eukaryotes, and the polycistronic conditions mostly see as in a bacteria like as in a prokaryotes. Okay, this is stand. So, for example, you take as this is a template stand, they having as in what? Genes as present. Let us in this gene the DNA coding for polypeptide. See here in this DNA their code here they see it is completely code for here they completely code for as in the different amino acids. Here they code for as in the different amino acids like. So the total their coding of the genes they are based on the structural gene, the transcription unit said to be as in a monocistronic. Only one kind of that genes will be the code, that is called as what? That is called as a monocistronic condition. Very important here. This one monocistronic condition you see is only for the eukaryote. The monocistrons is called as what? This monocistronic they present as in only for the eukaryotic cells. Let's see here what are the cistrons. Cistrons means they get uptake the signals from the in DNA. Okay, here cistrons. Let here what are the cistrons? Cistrons means they get signals. They get as what? Signals from DNA. Okay, so mostly monocistronic conditions we see that in eukaryotes, and then here the polycistronic the cistronic condition you see as in that is prokaryotic cells. You see in that where? In that prokaryotic cell, eukaryotes, and then that monocistronic polycistronic condition you see as in bacteria and then prokaryotes. Let's see here. When see after eukaryotes, the monocistronic structure genes having their interrupted coding sequence, the genes that eukaryote their splits. So when you observe, for example, here when you take as in one particular the gene, when you take as in the particular gene like, so in this gene they completely show as in split genes. In the monocistronic genes, here they are produced in the some split genes like. These are completely called as the split genes that get produced. Okay, let's the coding sequence they expressed they are defined as an exons. So where these regions are called as what? These are exons. These are completely called as what? Exons, because what? They are completely defined as a one particular coding sequence they are present here. They completely they get exons said to be as in a sequence of that completely the mature processor RNA. So in this total coding sequence, they are completely these exons they are having as a coding regions. Am I right? So these coding regions they are completely after to get joined with the help of the DNA ligase enzyme. After to get make as what? The processing mRNA will be formed. Completely the process mRNA will be get formed. And the exons are interrupted by as an introns. See here before this here between these mostly between these regions is called as what? These are completely introns. These are completely called as what? introns. In these introns, they are non-coding sequence. They are completely called as what? Non-coding sequence. Here, it does not code for any amino acids. Now, the introns or interrupted sequence do not appear as in a mature process RNA. The split genes arranged further, they complicate that in a their definition of the genes terms as in a DNA segment. So, after what happened here, for example, when you observe here, it is in a the coding sequence. It is in a coding sequence. It is also coding sequence. So then finally what happened in this coding sequence, they get bind with the help of the DNA ligase enzyme to get make as in one particular the mRNA strand will be get formed. One particular that mRNA strand mature here it is in a the mature mRNA form. Mature mRNA is formed. That process is called as a splicing process. That is called as what? That is in a splicing process. Let's see. Here, the inheritance of the characters also they are affected by as in a promoter and regulated sequence of the structural gene. So these all characters, the promoter characters and how where they get binding, the total they are affected by as in where on the structural gene. Hence, some this regulation sequence is loosely defined as a regulatory genes. So that purpose in these total genes are they get loosely they get bind with the completely mRNA strand. Even though this sequence do not code for any RNA or protein. So in these regions do not code for as any RNAs as well as any proteins. Okay, this is in a completely the process of in a transcriptional unit. Okay, this is. Let's we discuss about as any types of RNA and the process of transcription. Let's we discuss here. Let's we discuss here types of RNA, the process of transcription. First, let's see in bacteria, the three major RNAs here is present. One is in a mRNA that is called as a messenger RNA, and second one is in a tRNA that is called as a transfer RNA. The third one is ribosomal RNA that is called as an rRNA. So when you take as in any one particular that bacteria they contain as in how many RNAs is present? Three RNAs. One is in a that is a rRNA and mRNA and then tRNA and then tRNA. These three kinds of RNAs is present. Now, all these three RNAs are named synthesized by protein cell. So, in this total RNAs they synthesized by what? That in a completely in a RNA cell only. See in this RNA cell to get synthesized by as in a this all. Okay, like now this mRNA provides as a templates. Already we already discussed. For example, this in a one template strand. It is a completely what? One completely the template strand. The tRNA bind with as an amino acids and read the genetic code. So for example, here I am taking as in this is a template strand. Am I right? In this template strand, here has in the promoter site is present. Here they having as a terminator site is present. Am I right? It is in a promoter, it is a terminator, it is in a structural gene. It is in a the structural gene. Correct? Here he is saying but as what? The mRNA provides the template. So here, see it is there mRNA, the structural gene to get providing as what? In a mRNA. And tRNA bind with as an amino acids to get read that in a genetic code. Here, in this total, the promoter site, here see they having the number of here the codons as present. This total codons to get bind with the help of what? They bind with as a completely tRNA. They bind with as a tRNA. And the rRNA they play important role. They are structural. Here in this rRNA and they catalyst role during the translation process. So in this rRNA they participate as what? This total this both codons they get translated and after finally to get synthesis as a information. That is called as an already the ribosomal RNA. The ribosomal RNA function is what? It is to completely their synthesis. It is in a completely the synthesis of genetic information. Okay, that is in that process. The first the process of the synthesis of RNA. Once again, yeah, like here only. Next. Here, these are the single and DNA dependent RNA polymerase. They get act as a catalyst. This all type of RNAs in the bacteria. So here, the complete DNA dependent RNA polymerase they get act here. They are participate in this total process of means what the formation of the template and to bind with RNA sites and the translation process. Okay, the RNA polymerase bind with RNA protein. They initiate the transcriptional unit in this initiation process. So when they get adding here, this process is called as what initiation. In these use the nucleotide triphosphate in the substrate. So for example, see here in this the first promoter site. See this is a promoter na. So in this promoter site when they get uptaking here, their first one catalyst, that is what their RNA polymerase enzyme. That is what RNA polymerase enzyme, DNA dependent RNA polymerase enzyme that is having sigmoid factor. Okay, this process when they get entering, this is called as what the initiation process. That is called as what initiation process. After the initiation, what happened in this total mRNA? Say the first they formed as in that mRNA strand. Now what happened here? Now they get code will be they get read with the help of what in the tRNA. See in this mRNA they get completely get reading here, right? In this total part, it is a Hubble hypothesis, right? Here what happened? Here the number of hydrogen bonds will be they get removed. Means what? The number of peptide bonds will be they get removed. They get make as in one bulge shaped structure. That is in that in a process of in a mRNA. This is called as what? It is an elongation process. It is called as what? Elongation process. After elongation process, let's see here. Here the last one is the termination. So after when this in the splicing is takes place, means in the total in the sequence of the mRNA, in the sequence of the mRNA already we discussed what they having as an introns and exons is present. They having as an they are introns and exons. So here these are the completely these spots are called as exons, right? So in these exons to get completely to get bind with help of the bind with as with help of what in a DNA ligase enzyme, with help of the DNA ligase enzyme which to get form as in mature the DNA will be the comes sorry mRNA matured, the mRNA will becomes that is that is RNA polymerase rho factor, that is called as what RNA rho factor. That is a process completely in the splicing the last class we discussed. Let us here in this polymerase the template depending upon the fashion they are following here the mostly the polymerization of the template that depend the fashion the following the rule of in a complementary. So when they get followed here, when they get followed as in the rules and the complementaries in somehow the facilities here, somehow the facilitates opening of the helix and the continuous elongation. So once they get helical they get open, what happens they continually they get elongated and only one shot the straight the RNA remains will be there bind with as an enzyme. So for example any one site here only any one site they get start with as an they are binding, and once the polymerase they reach as a termination region their nested that RNAs will be follows. So after once they get enter here and after to get reach in this region that what happen the RNA will be they get fall down, and so also RNA polymerase this result as a termination. So when they get here the synthesis will be takes place when they getting as a matured mRNA, this is completely in a what it is a process of in a termination process. Okay, this is remaining to discuss in next class. Thank you for watching. Please share, subscribe and like.